Hello everyone. On behalf of School of Life Sciences, we welcome you all for the National Science Day 2024, organized in association with Tamil Nadu State Council for Science and Technology, Government of Tamil Nadu, and National Council for Science and Technology Communication, Department of Science and Technology, Government of India. I am Dr. S. Ranjani, working as Assistant Professor in School of Life Sciences, B.S. Abdur Rahman Crescent Institute of Science and Technology, Chennai. I'm going to present a topic on applications of nanotechnology in healthcare. Coming to the introduction, nanotechnology is a term used to define areas of science and engineering in which phenomena occurring at nanoscale dimensions, that is 10 to the power minus 9 meter, which have been used in design, characterization, manufacture, and applications of materials, structures, devices, and systems. The term nanotechnology was first introduced in 1959 when Richard Feynman presented a presentation on making a things at the atomic and molecular levels. Nanotechnology is the most promising technology, and researchers have investigated it as a novel technique in medical research because advancement in nanoscience have led to the emergence of a newly generated nanostructures. Each of them has a unique set of properties that account for the astonishing application in healthcare sector. Not only in healthcare sector, the nanotechnology has set its footprint in all the areas of science and technology. Nanotechnology can spur economic growth and enhance the capacity and quality in all the industrial sectors. It has substantially contributed to social welfare, society's development, and molded the nature of modern living system. Nanotechnology has a potential to significantly alter the societal dynamics as well as economic conditions and human life. What is the need for nanotechnology in the medical field? We know that healthcare is a basic human right has often become the focus for the development of innovative technologies. Technological process has significantly contributed towards high quality, on time, acceptable, and affordable healthcare. In the healthcare industry, nanoparticles led to several dramatic improvements because of their unique features, that is, catalytic activity, chemical stability, thermal conductivity, and optical properties. The inclusion of nanotechnology and its allied like nanocarriers, nanosystems in medicine is known as nanomedicine. Nanomedicine brings numerous health benefits in disease prevention, diagnosis, and treatment at the nano level. Incredibly, these smaller size particles facilitate and helps in the development of nano devices to demonstrate and explore properties and operations. Nanoparticles can be developed into suitable system according to the need with change in their size, shape, surface properties which can efficiently be utilized for imaging, diagnosis and treatment of serious diseases including cancer, cardiovascular disease, ocular disease, central nervous system related diseases. So currently, every researchers are using nanoparticles to target particular site or particular location to treat various systems, to treat various diseases. That is called as drug delivery systems, and it improve medical imaging also. Due to their unique properties, that is like ease of production at low cost and non-toxicity, bio-inspired nanoparticles hold a cutting edge over traditionally produced particles or molecules. So the benefits of nanotechnology or nanostructures are it can directly target the disease or cells, so it ultimately reduces the frequency which we are taking during our routine medication. What are the different types of nanoparticles are being used in the medical field? Nanoparticles are three different types: organic type nanoparticles, inorganic type nanoparticles, and carbon based nanoparticles. Under organic type nanoparticles, they are further classified into oxomes and polymers. These use polymeric materials for the development of organic nanoparticles, which includes polymeric nanoparticles, polymeric missiles, polymerosomes, dendrimer, nanostructured lipid carrier, solid lipid nanoparticles, liposomes, and nanoemulsion. The second category is inorganic type nanoparticles. They are further subclassified into metallic, metal oxide, and quantum dots. The inorganic nanoparticles comprises of metal-like nanoparticles which are made by metal precursors. They are in particular have unique optoelectrical properties. We use metals during the reduction and capping. So examples are silver nanoparticles, iron oxide nanoparticles, gold nanoparticles, calcium phosphate nanoparticles, quantum dots, and mesoferrous nanoparticles. The third classification is carbon-based nanoparticles, which comprises of nanotubes and fullerenes. So they are based on carbon materials. Coming to the various nanosystems that are used in healthcare sectors, they are carbon nanotubes, dendrimers, nanodiamonds, quantum dots, nanofilms, liposomes, nanofibers, metal nanoparticles, biodegradable polymer, carbon-based nanomaterials, hydrogen nanocomposites, and many more. The first one is carbon nanotubes. I would like to give a brief details about these type of different nanostructures that are being used in the healthcare sector. So carbon nanotubes are nano-sized, seamless tube which is made up of graphite sheets. So it is like tube-like structure. So one end is open, that is the one terminal part is open, the other terminal part is capped with fullerene. They have good mechanical strength and it is good absorber of magnetic radiation and heat. It can conduct heat and it can function by using a very good catalytic properties. These carbon nanotubes are included in the fullerene nanotube family because this is made up of fullerene system, that is the fullerene caps. The benefits of using these carbon nanotubes are they can cross partially permeable cell membrane very easily using the special mechanism because of nano insides. They can carry small organic drugs, molecules, proteins, peptides, nucleic acids, antibodies to the precise location. These carbon nanotubes can hold the materials or drugs by means of covalently at bonding or by means of adsorption or by means of encapsulations. These carbon nanotubes have wide application in X-ray imaging. These carbon nanotubes can withstand heat up to 158 degree Fahrenheit. So it cannot be easily destroyed by laser beam also. Hence, these carbon nanotubes can be effectively used in the treatment of cancer. The second one is dendrimer. We know that dendrimer is having a branched structure. So dendrimers are naturally biodegradable nanopolymer. They are macromolecule nanostructure. They are globular in shape because of the presence of branched layers. They are varying from different sizes ranging from 1 to 10 nanometer, and they can easily penetrate to the cell membrane. They can easily penetrate into the cell membrane by means of their lipophilic nature. Because of this lipophilic nature, they are the ideal system for the healthcare sector and it can be used in targeting several genes to the precise location. The dendrimer is a major nanocarrier system for the drug delivery purpose as well as it can be used for gene delivery purpose. So the dendrimer is made up of three important components. That is, one is the core which is present at the center and many functional groups are at the terminal of the branching loop, and it has many functional molecules which can be folded inside or it can be attached to the branching points. So these attachments are made possible by means of physical interaction or by means of chemical interaction. Physical interaction is nothing but just encapsulating the drug inside these hollow space. Chemical interaction is either by means of electrostatic interactions or by means of covalent interaction. Hence, the dendrimers are very important in carrying genes at targeted drug delivery. Coming to the next one, that is nanodiamonds. As a term nanodiamond, this nanostructure consists of single diamond crystal with a carbon in sp3 configuration. It is made up of pure carbon. The particle size approximately ranges from 4 to 5 nanometer. They are very hard, they are very chemically inert, and uh, they have very high thermal conductivity, and it is biocompatible in nature. They have tunable surface. These carbon particles, that is nanodiamonds, they are very tunable, and because of the large surface area, it can hold drugs and genes that can easily conjugate. So that is the major advantage of these nanodiamonds. Fluorescence can be produced by means of these nanodiamonds, which can be used for imaging probes or for diagnostic purpose, for diagnostic cancer can be used. So these properties made these nanodiamonds as an important nanostructure in the term of healthcare sector. So these nanodiamonds are also consist of two major components, that is inner diamond core, that is with carbon atom in sp3 configuration, and the outer graphite shell, which is having carbon in sp2 configuration, so that various functional groups can be conjugated terminally by means of bond formation. Uh, these nanodiamonds can be produced or synthesized by means of high temperature, high pressure, and chemical vapor deposition methods. So it is also made up of carbon in sp3 and sp2 configuration as a whole. So coming to the fourth one, that is quantum dots. These quantum dots are synthetic nanostructure which is ranging from 5 to 10 nanometer in diameter. They are semiconductor in nature. This semiconductor nature helps them to transport electrons during its function. So these quantum dots are made from heavy metals such as cadmium, and they are carcinogenic. Therefore, they cannot be widely used in healthcare sectors. However, graphene and carbon quantum dots are safe and stable that can be widely used in the healthcare sector. So instead of using cadmium, we can go for graphene and carbon that can be used for various diagnostic as well as treatment purpose. That is the advantage of quantum dots that can be synthesized by using various chemical. So we can leave the cadmium during synthesis when it is used in the healthcare sector, and we can use graphene and carbon for making it as safe. The next one is nanofilms. The nanofilms, as the term suggests, it is made up of polymeric sheet. So it is a nano, so it has a very large surface area and thickness of relatively few nanometers, that is 10 to 100 nanometer in diameter. So these nanofilms have oppositely charged layers. They are assembled together to form a multi-layer like ultra-thin biofilms. They forms layer by layer with different like opposite charged particles. So these layers are deposited one by one by means of deposition techniques. Various methods are used for depositing these sheets of individual layers by means of fluidic assembly, electromagnetic deposition, electrospin coating, and immersion. So this is also used for medical purpose, like preparing thin films or films to cure wounds. The next, coming to liposomes, we know that liposomes are spherical vesicles. So this is spherical vesicle made up of one or more lipid bilayer with an aqueous compartment which is located in between them. So they are found in variety of sizes starting from small to few nanometer. The size can be dictated as per the composition. They are capable of entrapping various substances including hydrophobic as well as hydrophilic. They are considered as a very efficient drug delivery system because it hold a number of materials to specifically reach the target sites. And another reason is that the composition is very similar to that of cellular membrane found in the body. So these liposomes interact with the cell membrane and it can easily deliver the drug to the target cells. So their larger size also enables them to deliver a high quantity of drugs because the size can be dictated as per the composition. So we can stuff lot of drugs or molecule inside these liposomes and we can make them to target the specific location. The major domains of healthcare in which nanotechnology, mediated nanosystems, liposomes mediated nanosystems are playing positive role in treating cancer. So this is about the liposomes. And nanofibers, they are like fine fiber like material which can be synthesized and which the size can be dictated by means of electrospin technology. These are also used in wide range of medical application. Depends upon the need. We can make the nanofibers and we can use it for curing wounds. Can be used for stitching and it can be used for sutures, etc. So metal nanoparticles plays an important role. There are different types of metal nanoparticles like silver nanoparticle, gold, platinum, palladium. So based on the needs, we can use metal nanoparticles to treat various type of diseases, like it is used to treat or break antibiotic resistance. It can be used in other wound healing purposes, prepared in incorporated bandages, cosmetic fabricated materials, etc. Biodegradable polymer, these are also made up of polymer which is biodegradable in nature. Carbon based nanomaterials like carbon nanotubes, nanodiamonds, quantum dots, everything will comes under carbon based nanomaterials because they are made up of carbon. And examples are fullerenes, buckyball structures, graphite. They are made up of carbon but in different forms like tubes, shapes, etc. Next is hydrogel nanocomposites. It is in the form of hydrogel. So it can be used as creams or gels to treat various diseases, or it can be used to target a particular site and to provide us a slow and sustainable release of drugs. This is all about the different nanosystems that are used in the healthcare sector. I would like to brief up the different systems that are used in various healthcare sectors. So far, we have seen about dendrimers, liposomes, quantum dots, missiles, nanosphere, nanoparticles, polymeric nanoparticles, and carbon nanotube. So these are the different applications in healthcare: gene therapy, drug delivery, cancer therapy, genetic disorders, ocular disease, cardiovascular disease, brain and central nervous system disorders. So gene therapy is a prime area where nanostructures and nanosystems are dedicatedly used in targeted gene delivery. Next one is drug delivery system. The site-specific targeting and delivering of drug is foremost essential for treating several diseases and disorders. Cancer therapy, this is very vast area where nanostructures have been used in diagnostic as well as in treatment procedures. Next is genetic disorders. Uh, these nanostructures are also playing important role in treating several genetic disorders, and these nanostructures are helping us to target the specific genes and drugs to the particular site so that the disease or the alterations can be corrected. Ocular disease, the eye is very sensitive area, even though nanosystems are playing crucial role in treating several eye diseases, that is ocular disease. Cardiovascular disease, this is one of the major diseases that most of the world is currently facing. So nanostructures and nanosystems are effectively used in treating several cardiovascular diseases. Brain and central nervous system disorders, this is very important organ of the human body. The nanostructures and nanosystems are very effectively working in treating several brain diseases and disorders. Coming to the different applications of nanostructures in healthcare. So nanoparticles, these nanoparticles are different types made up of different metals like silver, gold, copper, platinum, palladium, etc. They are effectively used as antimicrobials and antifungal agents, and they are effectively used in sensors, developing sensors. They are used as catalysts and they are effectively used for imaging purposes using diagnostics of cancer and various other diseases. Next is carbon nanotubes. These carbon nanotubes are tube-like structure that effectively deliver the drugs and molecules to the target cell. So they can easily used for delivering fibrinogen and bovine protein to the cells. These are the list of applications that are used in healthcare sector, and they can be served as a vector for gene delivery, as well as it is used in treatment of broken bones, that is fractures, osteoporosis, and it is effectively used in treating breast cancer also. The third one is dendrimers. We know that dendrimers is a branched structure which can hold drug by means of electrostatic or covalent interaction. They are used in targeting drug molecule to the particular site. They are used for diagnostic application also. They are used for gene delivery, antibacterial agents, anti-cancer drugs, and to improve vaccine formulation by acting as a carrier of antigen. Because of its branched structure, it can hold the antigens, and it is used in treating ocular diseases also. Nanodiamonds, as the name suggests, it is made up of single crystals of diamonds. They are used in treatment of bone disease by targeting the drug delivery, that is bone regeneration. They are used in imaging and therapy, and they are used in early detection of cancer, and it is used in the treatment of brain and breast cancers. Nanofilms, as I told, it is a film-like layer which has a various layers which is packed of oppositely charged particles. This acts as a useful biological, chemical, and nanomechanical sensor in electrochemical devices because of this wonderful electrical property. They are used for controlling release of the drug during drug delivery function, and they are used as nano patches, that is to heal wound. As we discussed already, it is made up of oppositely charged particles, and they are formed as films that are arranged layer by layer. So they can be prepared as a patches, and they can be used for treating wound that are caused after open surgery and to close incision. That is, the wound can be easily cured by means of applying these nanopatches. Liposomes. We know that liposomes are wonderful drug delivery system. They are used as drug delivery system because they can hold both hydrophobic and hydrophilic drugs, and it protects the drug from chemical and enzymatic degradation when it is entering into our body. And they have the ability to encapsulate anti-tumor drugs, for example, anthracyclines such as epirubicin, daunorubicin, and dox. So they can be easily holded inside the hollow part of the disc core part of the liposomes. So these are the different applications of different nanostructures that are currently being used in the healthcare system. One of the foremost important application of nanostructure in healthcare is by means of gene therapy. We know that gene therapy is a procedure to replace a defective gene in a DNA with a normal gene. The gene is usually inserted into the stem cell using a vector. Some stem cells are used because they have long life and they have a self-renewal ability, and therefore it can most suitable for targets for gene therapy. The vector used should be highly specific, and it should be efficient in releasing the gene or genes of variable sizes. It depends upon the genes that we are going to target. It should not be recognized as an antigen by our host immune system. The vector should have the ability to express the inserted gene. When the gene is correctly inserted into the gene, it inhibits and corrects the malfunctions of the mutated gene, and it induces the normal function. So this is the role of gene therapy when using stem cells. The other one is routinely used vector system is viral vector. These viral vector systems are used for years in gene therapy, and it is still being used. Uh, the prime use of viral vectors are they can insert into the host genome, they form a stable insertion, and there will be a long-term expression of the transgene. So this property is very much suitable for gene therapy. The most commonly used viral vectors are lentivirus, retroviral vector, adenoviral vectors. However, There are many disadvantages that are associated with using these viral vector. The few of them are generation of immune response. Our body can recognize it as antigen. So this may neutralize the effect of these viral vectors. Inflammation is one of the major drawbacks. This may lead to severe complication. And the third important thing is occurrence of off-target changes in the host body. So these are the major drawbacks when you use these viral vector. So when these virus enters into the body, it may trigger the immune response and it may become less effective. So this is the major drawback that we are facing by using these viral vector. Hence, these vector systems should be replaced, or we need to find the alternative system for transporting genes into the correct target site. So we are planning to use these nanostructures, that is non-viral nanostructures, as a safe as compared to therapy by using these viral vector. These are the different applications and different advantages because these nanovectors are non-viral structures, hence it is safe. They are less oncogenic and rarely it will trigger the immune response. Synthesis, that is preparation, is very easy. There is no risk of virus recombination. There is nothing to worry about the virus. We are not using any viral part or anything. So there is no need to worry about this virus recombination, and there is no limit of size of the gene to be loaded. So the most positive advantage of using these nanostructures are they are positively charged because we can fabricate the structure as a positively charged, and we can make it as a size nano in size that is suitable for the need. And because of its size and high surface to volume ratio, these nanostructures enables to penetrate deep into the membrane and making them ideal vectors for gene therapy. So these are the major advantage of using the nanostructures in gene therapy. The first and foremost thing is we can easily target a particular gene in the particular site without any complication. So these are the pictorial representation shows that this is the different type of nanosystems that are being used in gene therapy. The first one is liposomes. We know that liposomes can hold both hydrophilic and hydrophobic. They can hold a number of materials into it. So when the gene is inserted into the liposome, and it can prevent it from degradation to external environmental conditions or enzymatic activity. So when we are loading our gene inside this can be prevented from enzymatic attack or chemical attack. The second one is chitosan alginate nanoparticles loaded with growth factors. So these nanoparticles have a core structure which can load nanoparticles can hold a number of growth factors that can easily transport the growth factor into the target cell. The third one is super paramagnetic iron oxide nanoparticle, that is called as pions, with fluorescent molecule. When the fluorescent tag is attached, we can easily use it for diagnostic purpose. We can easily track the movement of the nanoparticles, and we can use it for diagnostic purpose as well as imaging purpose. And the fourth one is quantum dots. Quantum dots are made up of carbon particles that can easily be fabricated by means of gene that to be holded in, and it can target the specific site because of the smaller size and surface to large volume ratio. So when it is inserted into the stem cells, then we know that stem cells are easily differentiated and it is self-renewable. So these stem cells have different properties which will be helpful in angiogenesis, that is formation of new blood vessels. This can be used in the treatment of cancer, or it can be used in the treatment of cardiovascular disease. So based upon the need and based upon the treatment norms, we can use cells, and we can use the materials to target a particular cell. The first one is about the cell differentiation, where the tissue repair will take place. The stem cells will, with the particular gene, helps in the formation of new blood vessels and increase the paracrine secretion in angiogenesis. During angiogenesis, it helps in the secretion of paracrine. So it helps in the formation of new blood vessels. So next one is stem cells isolated from placenta. We know that placenta is folded with lot of beneficial effect. So when the stem cells are isolated from placenta, There will be a continuous release of growth factors from placental stem cells that improves the function of cardiac tissues at the site by means of releasing the drug slow and sustained release of these growth factors at the particular target, so that it can specifically target at the myocardial infarction site and leads to healthy heart. So these holds a number of growth factors. So this will slowly and released into the particular target site, and this helps in the formation of new T cells. With the help of these growth factors, they can target the particular site, and they can treat the myocardial infarction and leads to healthy heart. And the next one is the stem cells, which are binded with the spions, because it has fluorescent imaging property that will glow when it is exposed to particular radiation. So when there is internalization of spions by means of endocytosis, their glow will be detected by means of detector, and it can help in tracking the stem cells. The locations can be easily tracked. So these stem cell receptor will bind with quantum dots, and when it is entering into other membrane, that is by means of endocytosis or by means of diffusion, the internalization of quantum dots can be monitored by means of sensors that help in the easy tracking of these stem cells, and we can monitor the progress of the nanoparticles which is loaded with spirits. So this is the general application of nanotechnology in gene therapy. So based upon the disease and based upon the need, we can fabricate the material and we can load the gene to the particular target site. Even these fluorescent materials can be tapped to monitor and detect the movement of the particles. This helps in the tracking of particles along the way. So this is all about the nanotechnology in gene therapy. Next, we will see about nanotechnology in targeted drug delivery. We know that nanovectors have great potential in target specific drug delivery for the treatment of various diseases. Because continuous and controlled release of drug in desired amount, specific and localized drug delivery also reduces the drug doses. The smaller sized nanoparticles allow them to penetrate deep into the tumor cells, and thus they can be useful in improving the cancer treatment. Not only in cancer treatment, these nanoparticles are used in treating various diseases like Alzheimer's, Parkinson's, several ocular diseases we'll be seeing in forthcoming slides. These nanoparticles are used for drug delivery, must contain some important components. The three important components are particle core, the outer biocompatible protective layer, and linking molecule for increased bioactivity. These nanovectors can be modified according to our need. So before drug delivery, these modifications can be included over the nanovectors. These modifications can be included by means of coating with the ligands by using peptides, folic acid, and many antibodies are available for this. So based upon the need, we can fabricate the nanovector and we can design it. The ligands are nothing but they may be a peptide, a folic acid, or antibodies. They can be attached to the nanoparticles and they can bind specifically to the target sites to enhance the specificity even more than the normal one. So when the ligands are attached to the nanoparticles, they can specifically target to the targeted site. Since the nanovector possesses unique properties like size, shape, and physical qualities and chemical qualities, and various modifications can be performed during the drug loading to make it as a slow and sustained release of drugs in the target site. So this is the advantage of nanotechnology in targeted drug delivery. In previous slides, we have seen about the application of nanotechnology in targeted drug delivery system. In forthcoming slides, we will be seeing the application of different nanosystems in treating the particular disease. The first and foremost thing we are going to see about the application of nanosystem in treating the cardiovascular diseases. So, cardiovascular disease is one of the major problems that most of the people are facing around the world. We can see that it causes millions of deaths around the world. Various treatments have improved and survival rate of patients with heart diseases, but none of them have received complete cardiac regeneration, especially for the patients after cardiac infarctions. So we need to find some alternative measurements to come out of these hurdles. So one such alternative is the compatible nanosystems. These nanosystems are biocompatible and are efficient in transferring the genes to stem cells. So because of the structure, it can easily transfer the genes to the stem cells. The one such example, before two slides, we have seen that liposomes loaded with target gene and how it works. Similarly, the liposomes are one of the best carrier for gene delivery, as they can prevent the non-specific binding of the gene and protect from degradation. When the liposomes are enclosed, uh, there is no chance of the genes to get exposed to any enzymatic activity or chemical activity, so it can be safely travel into the barrier and it can reach the target site, and without any degradation it can reach the target site and bind to the specific cell. So this is how the liposomes work, and one such wonderful similarity is it is the liposomes are very similar to that of cellular membrane. Next is polymer. The polymers show improved specificity for targets and higher efficiency. So polymer is another nanostructures which will be helpful in treating the cardiovascular disease. And next is chitosan alginate nanoparticle. They were used for delivering growth factors in placental cells. Through placental cells, the growth factors are loaded, and when it is released into the particular site, there is a continuous release of growth factor. So it improves the function of the cardiac tissues at the site of myocardial infarctions. So the slow and sustained release of growth factors will help in the regaining of the function, and the tissues will work normally, and it helps in coming out of the particular site from myocardial infarction. And also, nanoparticles have the potential for tracking and monitoring the stem cells, that is, we call as pinoids. So, where the magnetics nanoparticles are loaded with some fluorescent emitting molecules, where we can track where the stem cells are moving, and it can be used for tracking also, as well as it is used in diagnostic purpose also. This is because the superparamagnetic ion oxide, that is pions, are made to enter into the cells by attaching to the cell surface. These cells are internalized by endocytosis. By means of endocytosis, these pions will enter into the system, and it works by releasing the particular gene or particular cargoes into the particular site and helps the patient to recover from the particular cardiac infection or cardiac arrest, whatever that we are looking at. Next good example is quantum dots. They are made up of carbon particles. So they can also be used for monitoring the live cells for a long time. So these systems are helpful in treating the cardiovascular disease, and many scientists are working in this area to come out of these cardiovascular disease. Another one, a good example is hypertension. Hypertension is a disease that gives rise to many problems, and they leads to myocardial infarction, heart failure, stroke, increased blood pressure, damage to many organs, including eyes, brain, kidney, etc. So many drugs have been used to treat these problems and complications, but the drugs have very short half-life, low bioavailability, poor solubility in water, and there are many side effects because of these drugs. So when these drugs are loaded in this kind of liposomes or chitosan alginate nanoparticles, these materials can be delivered into the target sites, and slow and sustained release will help in the release of drug to the target sites and helps the patient to recover from the infections or problems that they have faced. Nanocarriers have been used for treating hypertension. For example, lipid carriers are used, solid lipids, polymeric nanoparticles, liposomes, and nanoemulsions have been used. These are just few examples, but nanotechnology have very promising application in treating many cardiovascular diseases through these non-viral stem cell based therapies. So further researches are going on based on these nanovectors in the cardiovascular disease, as well as living models are used to find out the effect of these nanocarriers, and this will help to develop very good platform for the safely use in human beings. So this is about the treating cardiovascular disease by using nanosystems. It is nothing but we are loading the drugs and we are targeting into the specific site. Slow and sustained release will help the patient to take the drug very slowly, as well as the genes can be targeted to the particular sites. The expression of the particular gene helps to overcome the patient from particular disease that they are suffering. Next, we'll see about nanotechnology in the treatment of ocular disease. The efficient delivery of drugs in the eye is an enormous challenging target because of the presence of complex barriers and elimination of mechanisms in the eye. We know that there are several barriers in the eye, which includes tear film that is present over the surface of the ear, ocular surface epithelium, this is also present over the surface, and internal blood aqueous and blood retinal barrier. These are the different barrier systems that are present in the eye surface. So we need to find some alternative for the proper delivery of drug for treating particular ocular diseases. So nanoparticles are, however, able to overcome these barriers because of its smaller size, high surface to volume ratio, and highly variable surface properties. We can modify the properties of the nanoparticles to achieve the particular delivery, and so that they can effectively act on the target sites without any toxic effects. So these are the important points that we have to look into the treatment of ocular diseases, and these nanoparticles can effectively and efficiently transport the drug into the target sites without toxic effects. They are biodegradable also, which means that they do not require surgical removal after they have been delivered the drug. And there are two type of eye diseases. One is anterior eye disease, another one is posterior eye disease. The anterior eye diseases includes cataracts, conjunctivitis, keratitis, dry eye, and corneal injury. These are usually treated by using eye drops, but the corneal barrier causes drugs to have poor bioavailability. So the treatment rate will be keep on increasing, so that the drug will not effectively work on target cells. So what we have to do, we have to work with the nano system so that we can increase the bioavailability by prolonging the retention time of the drug on the surface of the eye and improving the penetration of the drug. This will help in clearing of the anterior eye disease. When we are working with posterior eye disease in the choroid or retina, which includes retinoblastoma, glaucoma, choroidal neovascularization, macular degeneration, and other posterior uveitis, interocular injections, and they causes severe side effects because these when we are injecting drug by means of interocular injections, there are many side effects. So we need to design a nanosystem that have improved the delivery of drugs into the anterior as well as posterior portion of the eye, and the various nanosystems used for the purpose, which includes nanovesicles, nanoimplants, nanoparticles, and hydrogens, are useful in the treatment of ocular diseases. As we see that ocular diseases are very sensitive, there are several barriers. This nanosystem effectively target the particular site and helps in clearing of the disease which are affected by anterior portion as well as the posterior portion. This is about nanostructures works in the treating ocular diseases. Next, we'll see about the applications of nanotechnology in the treatment of brain diseases. You know that brain diseases can be treated efficiently if we can overcome the issues of the blood-brain barrier. So various types of nanoparticles can cross the blood-brain barrier and effectively deliver the drug to damaged areas of the brain. We know that blood-brain barrier is a boundary between the circulating blood and the neural tissues of the brain. The presence of this blood-brain barrier is a major hurdle for the transportation of the drug to the particular part of the brain. So it does not allow the to enter into the central nervous system, and it maintains the homeostasis of the brain. So if there is any disturbances in the blood-brain barrier, that leads to some neuroinflammatory conditions, some neurodegenerative diseases like Parkinson's disease, Alzheimer's disease, etc. In some cases, even damaged blood-brain barrier also does not allow the drugs to enter into the brain. So the use of these nanoparticles like organic and inorganic nanomaterials can easily penetrate into the blood-brain barrier. Inorganic nanomaterials like silica, molybdenum, cerium, iron, and gold can easily enter into the blood-brain barrier by means of its unique features like smaller size, large surface to volume ratio, their surface charge, whereas the organic nanomaterials can include PLA, PLGA, and tree halos for the faster uh, entry into the blood-brain barrier, as well as bioavailability, as well as slow and sustained release of the drug into the target cell. The distinctive feature of these nanoparticles are able to treat several neurodegenerative diseases um, because of its unique nature. It can easily penetrate into the cell membrane, that is blood-brain barrier, and it can effectively work on the target site. There will be a slow and sustained release of the drugs. Some nanoparticles by themselves have some therapeutic effect. For example, antioxidant activity. So they reduce the ROS levels and helps in the faster recovery of the particular tissues while undergoing for the treatment. So while we are using nanoparticle as a delivery system, these nanoparticles will be conjugated with some ligands in order to improve its performance. They will interact with these ligands and they will penetrate into the blood-brain barrier, and they will cross the blood-brain barrier and enter into the target cells. These nanoparticles can adopt multiple pathways for the entry into the blood-brain barrier. So these are the different pathways that has been represented in the figure, where we can see the passive diffusion, transmembrane efflux, paracellular transport, receptor-mediated transcytosis, carrier-mediated transport, adsorptive-mediated transcytosis. These are the different methods where nanoparticles will take up and enter into the blood-brain barrier. So they will cross the blood-brain barrier and they will act upon some neurons or active astrocytes of the brain. In receptor carrier mediated and adsorptor mediated transcytosis, the penetration of nanoparticles across the blood-brain barrier and various ligands are involved, such as the ligands that can absorb protein from the bloodstream. So this is like adsorptive mediated transcytosis. They will absorb some protein from the bloodstream. And second one is the ligands that can directly involve with the blood-brain barrier transporters or receptors. This is the way. So they will directly interact with the transporters or receptors so that they can easily travel the blood-brain barrier. Some ligands can increase the hydrophobicity and charge of the nanoparticle. Based on the charge also, they can easily penetrate and they will enter the blood-brain barrier and reach the target site. Some ligands can improve the circulation time of the nanoparticles in the blood. This will slow and steadily allows the nanoparticles to cross the blood-brain barrier and enters into the target cells. So the morphology, surface charge of the nanoparticles plays an important role. We call as zwitterions, neutral nanoparticles. These kind of nanoparticles will have greater circulation time when compared to positive or negative charged nanoparticles. So once these nanoparticles overcome the blood-brain barrier, and these nanoparticles can be used in the treatment of several neurodegenerative diseases, as I mentioned before, like Parkinson's disease, Alzheimer's disease, and many other. So this is how the nanoparticle work. So all the nanoparticles will not follow all the pathways to enter into the blood-brain barrier. According to the particle size, shape, physical chemical characteristics of the particles will dictate either in the method of the diffusion or efflux or paracellular transport or receptor mediated, carrier mediated, adsorptive mediated to cross the blood-brain barrier and acts upon either neurons or on the cells which are responsible for the particular disease. This is how the nanoparticles will act upon and functions in the treatment of brain diseases. Next, we will see about the application of nanotechnology in cancer diagnosis and treatment. We know that nanomedicine involves the implementation of nanotechnology in the treatment, screening, and diagnosis of various cancer. So we can categorize into different strategies how for the nanotechnology is applied in cancer treatment and diagnosis. They are based on utilization of different methods involving nanotechnology in cancer diagnosis, methods used for cancer treatment, targeting the cancerous microenvironment, targeting the drug-resistant tumors, personalized therapy for cancer treatment through thermal ablations. So these are the different strategies, and I'm going to tell you, or I'm going to brief you about each and every strategies that are followed in treating and diagnosing the cancer. The first one is utilization of different methods involving nanotechnology in cancer diagnosis. So, when we speak about diagnosis, there are obstacles in early detection of various types of cancer. These can be expected to be solved by using some nano systems like nanotubes, nanocantilevers, nanoparticles based probes, and nanowire arrays. And these nanosystems will help in the diagnosis of cancer at early stage. And there are several researches were going on to develop technology or strategy in the early diagnosis of cancer. So, on the basis of fluorescence resonance energy transfer, these nanoprobe systems have shown the potential to detect DNA mutation in tumor cells as well as to detect the loss of DNA. So, these are the advancements were followed and implemented in the diagnosis of cancer by using nanotechnology. So this is like this field is keep on growing, and the system has been keep on updating by applying novel and innovative technology to rapid diagnosis of cancer at the early stage. The second one is methods used for cancer treatment. So there are a number of methods are currently available to treat cancer. To is the intake of vehicles into the target cells. So where we are using some vehicles into the target cells, number of cancer targeting ligands such as growth factors, folates, cytokines, antibodies have been used. By means of using these nanoparticles, we can enhance the permeability and the retention time of the nanoparticles, which has been loaded with some drug. For example, caplostatin, and many some drugs are under study to find the retention time as well as bioavailability of the particular drug in the target cells. This is how different strategies are developed to treat the cancer by using these nanosystems. And there are several strategies are available. Apart from that, we are coupling metallic nanoparticles with some ligands, and we can couple some oligonucleotides with our vehicle, as well as we can use quantum dots and magnetic uh, doped metals, nanoparticles, and gold nanoparticles. These are very, very important, and they have a special features to enter into the cell and release the drug in a slow and sustained way. So, the direct observation of circulating tumor cells in blood, there are some new strategies has been developed to monitor the cells which are circulating in the bloodstream. So this is how the nanoparticles and nanosystems helps in the treatment of cancer. Uh, Still, there are several methods have been developed, and still several methods are under study to treat the cancer in various possible ways. And third one is about targeting the cancerous microenvironment. So this is an important platform. One such example is like we can use arginine, aspartic acid, and glycine motif has been found to display on strong selectivity and affinity for the cell surface in many proteins. Therefore, it is an appropriate ligand for therapeutic nanoparticle targeting the cancer cells. So, this motif plays an important role, and the motif has been designed to target the particular site to target the cancerous cells. These nanoparticles are in the form of prodrug or nanoformulation. The drug that has a short life or that is extremely cytotoxic in circulation may now can be controlled because of this low and sustained release. Though tumor specific molecules, these drugs are targeted at the tumor microenvironment. This tumor microenvironment eases its transformation into an active state upon arriving at its destination. By attacking some stroma or by attacking the tumor cells, this tumor activated nanoformulation therapy performs its function. This is how the nanoparticles target the cancerous microenvironment and works very efficiently. So there are some nanoparticles which are available where we can say, for example, poly-based nanoparticles. Uh, these are under study in order to uh, study more on cancerous vasculature and to increase the transfer of drug into the cancerous sites. So this is how the transfer targeting the cancerous microenvironment work. And the next important strategy is targeting the drug resistant tumors. So we know that there are several drug resistant tumors. For example, metastatic colon tumor cells, for example, that can overexpress some protein which is called as integrin. They are found targeted by pegylated liposomes. Okay, these pegylated liposomes alters with the fibronectin mimetic peptide. So this particularly target the particular overexpressing integrate, and it causes some changes over the cell. And nanoparticles are made to increase endocytosis when targeting this multi-drug resistant tumors, and to bypass or to efflux out of these pumps on the membrane, and considering various methods of drug resistant cancer cells. So this is a small example. This metastatic colon tumor can be targeted by pegylated liposomes. And next is personalized therapy for cancers. So this strategy uses paramagnetic nanoparticles. These nanoparticles are used to study angiogenesis, uh, as well as to study the nascent melanoma cancers. So these effectively control the cancer through early detection. So to target the malignant tumors with greater specificity and affinity, these nanoparticles are associated with some ligands. For example, small molecules, peptides, and monoclonal antibodies can be employed. So this is how the personalized therapy for cancer has been developed. So it varies from one cancer to another cancer and depends upon the site, target location, and type of nanoparticles that we are going to fabricate. So everything depends upon the cancer as well as the nanoparticles. Based on that, we can give the personalized therapy for particular cancer. The next is cancer treatment through thermal ablation. We know that nanoparticles can withstand high temperature. So these metallic nanoparticles show capacity for implementation in targeted hyperthermic therapy. At high temperature, it can withstand. For example, like carbon nanotubes, gold nanoparticles, silica nanocells, iron oxide nanoparticles. These nanoparticles withstand high temperature. Then these iron oxide nanoparticles are greatly employed as a therapeutic as well as diagnostic nanoscale agents in several cancer treatments. An increased amount of gold nanoparticles could be tagged with antibodies in opposition to specific tumor cells, and we can target the particular gold nanoparticles to the particular cells. So in advanced stage, the application of radio frequency fields to cells causes confined heat and death of tumor cells after the incorporation of nanoparticles. When it reaches the target sites, when you apply some heat, this will kill the particular cells which is lying near the nanoparticle. This is how these thermal ablation techniques works. So this application of nanotechnology in cancer diagnosis and treatment is very fast. I tried to give up most of the data in different forms, like utilization of different methods and methods used for cancer treatment and targeting the microenvironment, targeting the drug resistant tumors, personalized therapy for cancer, and thermal ablation techniques have been implemented to treat the cancer by using nanotechnology strategy. Next, I would like to summarize the application of nanotechnology in the treatment of several genetic disorders. So, we know that genetic disorders are caused by disturbance in the blood-brain barrier and several other factors are responsible for several genetic disorders. So, here are the few examples like Alzheimer's disease, where solid lipid nanoparticles, PLGA nanoparticle conjugated with Tet1 peptides are used, and it has implemented its function in inhibiting A beta aggregation and improved cellular uptake. Similarly, in Parkinson's disease, we use retinoic acid nanoparticles. This increases the viability of the loaded particles, that is, loaded compound, prevented apoptosis, and causes oxidative stress and toxicity in the particular cells. And next is amyotrophic lateral sclerosis. So here the calcium phosphate lipid coated nanoparticles are used. This can be able to pass through the blood-brain barrier, neutralize the RNS and ROS reaction. Next is Huntington disease. Curcumin, SLN, trihalose loaded zwitterion nanoparticles have been used. I told zwitterion are the best nanoparticle to cross the blood-brain barrier in the previous slides. So this reduced the activity of intermediate complex 2 and inhibited amyloid and polyglutamide aggregation. Next is cystic fibrosis. Here the lipid nanoparticles and PLGA nanoparticles coated with PEG are used. This increases the amount of membrane localized CFTR as well as effectively deliver the anti-inflammatory compounds. This is how the nano system works and efficiently uh, crossing the blood-brain barrier and actively targeting the target site which is responsible for the particular disease. Next, we'll see about the application of nanotechnology in the treatment of nervous system diseases. Fortunately, these nanoparticles are not only a valuable tool for genetic or neurodegenerative disorders, they have also been manipulated to treat several other neurological complications or traumas such as post-stroke neuroprotection and spinal cord injuries. So these are the major problem or complex areas, but studies have proven that nanotechnology can help to fight these severe diseases. Uh, the main causative agent or fact is that regeneration or repair in the central nervous system is another problem because we can't regenerate or restore because the damaged axon lack the ability to regenerate and regrow. It is very difficult task. There are several extrinsic factors which are responsible to inhibit the regeneration of axon. There may be because of age factor or because of some inhibitor molecules which prevent the regeneration of axon. In this regard, nanotechnology has taken into this step to found that it is very effective and potential tool to treat such disorders caused by damage of axon. Nanoparticles can help to regenerate or it can help to regrow the axon. For example, in the treatment of spinal cord injuries, many drugs are used, but these drugs have several side effects. When it is administered systematically, it is found to be highly inefficient because it is rapidly metabolized before it is entering into the target site and it is cleared off from the bloodstream. Now, the ultimate aim is to modify the drug by using some nanocarriers and increasing the bioavailability of the drug. For this purpose, we have conjugated, the paper has reported that they have conjugated adenosine with lipid squalene into nanoassemblies, and the experiments has proved that the neurological deficit score was improved and early motor recovery in the hind limb was observed. So this proves that nanotechnology has proven its potential to regenerate the axon. And a second good example is polymethyl methacrylate nanoparticle, which is used to target specific cell population of macrophages to decrease the inflammation without exhibiting any toxicity. We know that macrophages have been observed to perform a key role in entire inflammation process of microglia. These macrophages contribute to the chronic phase of neurodegenerative diseases, and hence they can be explored for better therapeutic targets. Hence, this polymethylacrate has been studied and it has shown that it decreases the inflammation caused by macrophages and it does not exhibit any toxicity for this. The next strategy is charged surface and surface pegylation. This enhances this process, allowing the cellular uptake rapidly as well as slow and sustained release of drug into the target site. So this is how the nanotechnology plays an important role in the treatment of several nervous system diseases. So here I would like to show the application of nanotechnology in the treatment of nervous system diseases. This has been taken from the paper from Anjum et al. 2020. So here we can see that these are the different diseases like optic nerve injury, glaucoma, spinal cord injury, stroke and spinal cord injury. So these associated axon damages, and these are the different nanosystem designed to treat these diseases, and these are the different target sites like the site of acute injury in axon. Here it is chronic optic cataract in case of optic nerve injury. Here it is case of rat optic nerve crush injury, retinal ganglion cells. These are the target. So how it is effectively helped in the regeneration and restoration of function of axon. This is how the nanotechnology helps in the treatment of several nervous system diseases. With this, I would like to conclude the session by summarizing the whole content. So far, we have seen the different types of nanoparticles like metallic, non-metallic, organic, inorganic, carbon-based nanoparticles, and we have seen about their therapeutical strategies in healthcare, in gene therapy, drug delivery, cancer therapy, genetic disorder, ocular disease, cardiovascular disease, treating several brain disorders and several central nervous system disorders. So, with these views and points, I would like to conclude that nanotechnology is one of the growing field, and it has been grown exponentially within last few decades. That we can really feel that, and the whole approach is focusing towards the healthcare sector, and it is increasing parallelly, and it is keep on increasing towards new discoveries, new findings in all the areas of health sector as well as other field also. In case of healthcare sector, it has been parallelly growing in diagnostic part as well as in the treatment part. So one of the major obstacles that we can face by using the nanotechnological approach in living systems for healthcare is its toxicity. So the toxicity study should be explored properly, and it should be clarified that it is non-toxic to the human beings before developing into the product for commercialization. So, coming to the conclusion, I can say that nanotechnology will push forward to develop more promising therapy to cope with more diseases, as well as it will provide the researchers with effective tools to actively solve the various bottlenecks that we are facing in the healthcare sector. Before winding up this session, I would like to share the major tech giants and startups in the nanotech healthcare market. So these are the different companies which are actively participating in producing some nano-based products in the healthcare sector, like biosystems, nanocareers, taxoncure, and they are playing the major role in treating several diseases, including cancer, several brain-related diseases. This is how the nanotechnology stands first, and it's playing its vital role in the healthcare sector, and hope the research will continue. More and more research will help to find more and more product in the welfare of human beings. Thank you.